December 19th, 2025. Mark it on your calendar. An interstellar visitor named 3I Atlas will make its closest approach to Earth, and it is carrying something you need to know about. Methanol, the molecular ancestor of RNA, and hydrogen cyanide, the chemistry they use in execution chambers, both molecules streaming off the same object, heading in our general direction. Harvard astrophysicist Avi Loeb poses the question that should keep you awake tonight. Is this a friendly gardener arriving to plant the seeds of life across our solar system? Or is it a serial killer dusting our planet with poison? The ALMA telescope has given us data that supports both interpretations, and that ambiguity is where the real story begins. Stay with me until the end, because what happens on December 19th might redefine what we think is possible in this universe. The chemistry is already on its way. If you find yourself as captivated by these questions as I am, consider subscribing so you can follow along as December 19th approaches and the story continues to develop. Leave a comment telling me where you are in the world right now, what hour you are listening to this, and whether you think 3I Atlas is exactly what the astronomers say it is, or something we have not yet fully understood. Part 1. The Payload. Bombshell. ALMA's methanol surge and the two-valve problem. Methanol is the chemistry of life. Cyanide is the chemistry of death. And 3I Atlas is shedding both. Let me say that again, slower this time, because the weight of it needs to land. An object that originated beyond our solar system, tumbling through the void for who knows how long, is currently spraying a cocktail of molecules into space that reads like a recipe for both creation and annihilation. The Atacama Large Millimeter Array, ALMA for short, has been watching this thing since late summer of 2025, and what it found should make every person on Earth stop and ask a simple question. What exactly is this thing doing? The timeline matters here, so let me give it to you clean. August 28th, 2025. ALMA detects methanol, chemical formula CH3OH, streaming off 3I Atlas. September 18th and 22nd. More methanol detection, stronger this time. October 1st. Methanol again, consistent and powerful. Now rewind slightly. September 12th and 15th. ALMA picks up hydrogen cyanide, HCN, the same poison used in gas chambers, emanating from the same object. These are not vague impressions or controversial interpretations of fuzzy data. These are spectrographic measurements, the kind of evidence astronomers trust with their careers. The molecules are there. The question is why. Production rate ratios tell us something is deeply wrong here. In typical cometary chemistry, you expect certain molecules to appear in predictable proportions based on how ice sublimates in sunlight. Think of it like baking. If you follow a recipe, the ingredients come out in roughly the same ratios every time. But 3I Atlas is not following the recipe. Measurements show methanol to hydrogen cyanide ratios hovering around 124 in some observations, 79 in others. To put that in perspective, most comets we have studied show ratios nowhere near this extreme. There is a precedent, one lonely precedent, a comet called C/2016 R2, which also had bizarre chemistry. But that was an oddball. This is an interstellar visitor doing the same thing, and that coincidence should unsettle you. ALMA did not just detect these molecules, it mapped where they are coming from, and that is where the story gets mechanical. Hydrogen cyanide appears to be venting from the nucleus itself, the solid core of the object, what we might call the hull if we were feeling speculative. Methanol, on the other hand, is forming far from the nucleus, more than 258 kilometers away, out in the plume where molecules are supposed to break down, not build up. This is the two-valve hypothesis. One valve on the hull spewing cyanide, another valve, or perhaps a distributed chemical process, generating methanol in the wake. If you were designing a spacecraft with two separate exhaust systems, one for propellant and one for cargo, this is exactly the signature you would expect to see. Standard sublimation models, the ones we use to predict how ice behaves when sunlight hits it, cannot reproduce this pattern. Normally, volatiles like methanol should sublimate symmetrically or enhance on the sunward side where heating is strongest. The models follow power law scalings, mathematical relationships that describe how production rates change with distance from the sun. Typical comets fall somewhere between -2 and -4 on that scale. 3I Atlas clocks in at -5.2. That number is an outlier. It suggests something other than simple ice melting is happening here. We will unpack what negative 5.2 actually means in a later part, but for now, just know this. The object is behaving in a way that our textbooks do not cover, so here is the narrative tension that should keep you awake tonight. Exhaust versus cargo. If one molecule looks like propellant residue and the other looks like biological feedstock, what does that imply? Imagine a spacecraft, and I am being deliberately speculative here, spewing reaction mass out one end to adjust its trajectory, while simultaneously jettisoning containers of prebiotic chemicals out the other. That is the image ALMA data conjures, whether we like it or not. Methanol is a precursor to formaldehyde, which leads to ribose, which is a sugar backbone for RNA. It is also the favourite meal of methylotrophic bacteria, microbes that eat alcohol for breakfast. Hydrogen cyanide, in the right doses, can participate in amino acid synthesis and even trigger seed germination in plants. In the wrong doses, it shuts down cellular respiration and kills you. Both molecules, together in this configuration, paint two completely opposite pictures. Now, before you accuse me of jumping to alien megastructures, let me give you three natural explanations. First, heterogeneous nucleus composition. Maybe 3I Atlas is a cosmic layer cake, with different ices frozen in different regions, and as it tumbles and heats, different layers expose themselves and vent different chemicals. Second, clathrate structures. These are molecular cages where one molecule traps another inside an ice lattice. When clathrates break apart, they can release trapped gases in bursts that look unusual. Third, thermal processing. Maybe the object spent time near a star in its home system and underwent chemical reactions that baked methanol into its structure, while leaving cyanide in volatile pockets. All three of these are plausible. None of them fully explain why the sourcing is so asymmetric, why cyanide comes from the core and methanol appears far downstream. ALMA has limits, and I need to be honest about them. The array can only observe when the object is at certain angles relative to the Sun, and it cannot see everything at once. There are data gaps, periods when 3I Atlas was 2, close to the Sun in our sky for ALMA to track it safely. The resolution is remarkable but not infinite. It can map the near-field coma, the region close to the nucleus, but the full heliospheric wake, the trail stretching millions of kilometres behind the object, is beyond its reach. So when I say ALMA found a symmetric venting, I mean it found a symmetry in the regions it could observe. The full picture is still developing. December 19th is coming. The geometry of that flyby matters more than distance alone, and we're going to show you exactly why Earth might still taste the wake, even from 270 million kilometres away. Part 2, the December 19th flyby, the chemical geometry that nobody is talking about. Closest approach to Earth happens on December 19th, 2025. 1.8 astronomical units, that is roughly 270 million kilometres, and NASA and the European Space Agency have been very clear about one thing. There will be no impact. The object is not going to hit us. Its trajectory is well understood, and the math is solid. But impact is not the only way an object can affect a planet. Let me explain why geometry matters more than distance in this case. Imagine Earth as a marble and 3I Atlas as a truck driving past you at highway speed, except the truck is leaking a fine mist out its back end. The truck does not hit you, but if you are standing downwind, you are going to breathe some of that mist. Now scale that up to astronomical units, and add solar wind, magnetic fields, and charged dust particles, and you start to see the problem. Or the opportunity, depending on your perspective. The question is not whether 3I Atlas will crash into us. The question is whether Earth will pass through any portion of the chemical wake it is leaving behind. Orbital geometry is three-dimensional, and most of the diagrams you see in news articles flatten it into 2 dimensions, which hides the complexity. 3I Atlas is on a hyperbolic trajectory, meaning it is moving fast enough to escape the solar system entirely after this flyby. Its orbital plane is inclined relative to Earth's orbit, so the two paths do not lie in the same flat sheet. Instead, they are skewed, like two highways crossing at different elevations. But the wake is not a thin line. It is a cone, a plume, a diffuse cloud of molecules and dust spreading outward as the object moves. The solar wind pushes on that cloud. The interplanetary magnetic field threads through it. And depending on how those forces align, portions of the wake can drift far from the object's geometric trajectory. ALMA found that hydrogen cyanide is depleted on the sunward side of 3I Atlas and enhanced elsewhere. Methanol shows the opposite pattern, stronger in certain directions, weaker in others. This is sunward asymmetry, and it tells us the venting is not random, it is directional. If you imagine the object as a sprinkler, it is not spraying evenly in all directions. It is pulsing jets from specific locations, and those jets are pointed somewhere. The question we need to answer is simple. Does Earth cross the methanol-rich sector, the cyanide-rich sector, or some blend of both? Here is where things get interesting and slightly unnerving. Charged dust and nanograins, particles so small they are almost molecular, can couple to the solar wind and the interplanetary magnetic field. When that happens, they stop following ballistic trajectories and start hitching rides on magnetic field lines. Think of them as microscopic rafts floating on invisible rivers. Those rivers can curve, twist and redirect particles in ways that pure gravity and momentum cannot explain. We have seen this happen with volcanic plumes on Earth. Ash and aerosols get caught in upper atmospheric currents and travel thousands of kilometres downwind, far beyond where simple physics would predict. The same principle applies in space, except the currents are made of plasma and the distances are measured in astronomical units. Let me give you some numbers, rough numbers, back-of-the-envelope calculations, but numbers nonetheless. If 3I Atlas is releasing methanol and hydrogen cyanide at the rates ALMA measured, and if those molecules expand into a spherical shell as they move away from the nucleus, then by the time they reach Earth's orbital distance, the concentration will have dropped by a factor of roughly 1 trillion. That is dilution on a cosmic scale. To reach lethal concentrations of hydrogen cyanide in Earth's atmosphere, you would need the object to release amounts that are orders of magnitude higher than what we are seeing. So the mainstream position, and it is a reasonable one, is that dilution kills the risk. By the time any of this reaches us, it will be so spread out that it is functionally irrelevant. But, and this is a big but, that assumes uniform dispersion. It assumes the wake spreads evenly in all directions and does not get concentrated by magnetic funneling or solar wind channeling. If a coronal mass ejection from the Sun happens to align with Earth's magnetosphere at the same time 3I Atlas passes, you could get a temporary filament, a narrow stream of particles nudged in our direction. The odds of that are low, but they are not zero, and if it happens, the concentration in that filament could be much higher than the average dilution factor suggests. This is speculative, I want to be clear about that. But it is not physically impossible. ALMA's angle-resolved spectra show directional biases in the molecular emission. Certain velocities, certain Doppler shifts, are stronger than others, which means the molecules are moving preferentially in specific directions. But ALMA maps the near field, not the full wake. It tells us what is happening within a few 100,000 kilometres of the nucleus. It does not tell us what happens 10 million kilometres downstream, or 100 million kilometres downstream where Earth will be. That is the observational gap we are living in right now. Solar activity is unpredictable on short time scales. A coronal mass ejection can erupt with a few hours' notice, or no notice at all. If one happens in mid-December, and if the magnetic field geometry is just right, particles from 3I Atlas could be shepherded along field lines in ways that defy simple ballistic models. I'm not saying this will happen. I am saying it is a scenario worth considering, especially when the chemistry involved includes both a precursor to life and a cellular poison. So December 19th is not just a date on a calendar, it is a geometric event. It is the moment when Earth's orbit intersects the projected zone where 3I Atlas's wake might be drifting. Whether anything actually reaches us depends on factors we cannot fully predict yet. Solar wind speed, magnetic field orientation, the exact release rates and velocity distributions of the molecules ALMA detected. We are flying partially blind here, and that should make you uncomfortable, or curious, or both. Next, we're going to talk about what those molecules actually mean, and why the chemistry itself fuels two diametrically opposite stories. Friendly gardener versus serial killer. Both interpretations are sitting on the same data, and that is where things get truly strange. Part 3. Friendly gardener versus serial killer. The chemical signatures behind the narratives. Avi Loeb, the Harvard astrophysicist who brought you Oumuamua as a potential alien light sail, has a new thought experiment, and it involves 3I Atlas. He frames it as a question of intent versus natural accident. Is this object a friendly gardener arriving to seed our solar system, with the building blocks of life, or is it a serial killer dusting Earth with poison? The ALMA data, frustratingly, supports both narratives. Let me show you how. Methanol is an astrobiological superstar. If you want to build the molecules of life from scratch, methanol is one of the best starting points you could ask for. It oxidizes into formaldehyde under the right conditions, and formaldehyde is a key ingredient in the Famose reaction, a chemical pathway that produces ribose, the 5-carbon sugar that forms the backbone of RNA. Ribose is not optional in biology as we know it. It is fundamental. Methanol is also the primary carbon and energy source for methylotrophic bacteria, microbes that thrive in environments where most other life would starve. If you wanted to kickstart prebiotic chemistry on a sterile planet, or give a nascent biosphere a metabolic boost, dumping methanol into the environment is a very efficient way to do it. Laboratory experiments have shown that methanol, when exposed to ultraviolet radiation or energetic particles, can trigger cascades of reactions that build increasingly complex organic molecules. Sugars, amino acids, nucleotide precursors, all of it starts with methanol. Hydrogen cyanide, on the other hand, has a reputation problem. It is the stuff of spy novels and execution chambers, a molecule that binds to the iron in your cell's cytochrome oxidase enzymes and shuts down your ability to use oxygen. In bulk, it is lethal. But, and this is where the dual nature comes in trace amounts, hydrogen cyanide is not a poison. It is a participant. Cyanide chemistry is involved in the Strecker synthesis, one of the primary pathways for making amino acids under prebiotic conditions. It also shows up in seed germination triggers for certain plants. The dose makes the poison, as the old saying goes. A little cyanide in the right place at the right time can be beneficial. A lot of cyanide in the wrong place is catastrophic. The difference is concentration, timing, and context. So here is the dual function delivery hypothesis, and I want to mark this clearly as speculative. What if both molecules are intentional? What if hydrogen cyanide is there to clear ecological niches, to suppress existing microbial competitors, or sterilize certain zones, while methanol is there to feed the colonizers that follow? It would be a 1-2 punch. First, you disrupt the existing equilibrium, then you provide resources for the organisms you want to establish. Alternatively, and this is the natural explanation, both molecules could be byproducts of the same chemical reaction cascade happening on the object's surface. Irradiation of organic ices can produce both methanol and hydrogen cyanide simultaneously. No intent required. Just physics and chemistry doing what they do. Let me give you a table, a comparison, in narrative form. Natural Comet model. Expected observables would include symmetric or slightly asymmetric venting, production rates that follow known sublimation curves, and molecular ratios consistent with other comets. ALMA's maps partially match this, but the extreme methanol to hydrogen cyanide ratios and the spatial separation of sources are harder to explain. Engineered tanker model. Expected observables would include precisely controlled release rates, directional venting that does not correlate with solar heating, and possibly non-gravitational accelerations as the object adjusts its trajectory. We do not have confirmed evidence of non-gravitational accelerations yet, but we also have not ruled them out. Hybrid model, natural body with catalytic surface chemistry. Expected observables would include unusual molecular ratios, steep power law dependencies on solar distance, and time-variable production rates. ALMA found all of these. So the hybrid model, where a natural object undergoes chemically enhanced reactions on its surface, might be the best fit for the data we have right now. If this is panspermia, why both molecules? If it is a toxin delivery, why so much methanol? The gardener carrying weed killer analogy is cute, but it does not resolve the ambiguity. It just highlights it. You could argue that a sophisticated seeding operation would include both enabling and disabling agents to maximize the chances of successful colonization. You could also argue that nature produces complex chemical mixtures all the time, and reading intent into them is a category error. Both positions are defensible with the current data, and that is the problem, or the opportunity, depending on whether you are a scientist or a storyteller. Detectability of biological payloads is another issue entirely. If 3i Atlas were carrying spores, dormant microbes, or complex organic polymers, we would not see them in ALMA's spectra. ALMA detects gas phase molecules, not solid particles, and certainly not microscopic biological structures. The imaging we have from optical telescopes shows a fuzzy blob, not a detailed surface. Even if there were macroscopic structures, tarps, panels, hexagonal grids, we would not resolve them at this distance with current instruments. In situ sampling would change everything. A probe that could fly through the coma and collect dust grains, analyse their composition, sequence any organic polymers, that would give us definitive answers. But we do not have that probe. We have ALMA, and ALMA gives us chemistry, not biology. So here is the measured verdict. Science needs more data. ALMA has given us a chemistry puzzle that is genuinely uncanny, genuinely strange, but extraordinary inferences require extraordinary proof. The data supports multiple interpretations, and until we can eliminate some of those interpretations with new observations, we are stuck in the ambiguity. That is not a satisfying answer, but it is an honest one. What we know for certain is that 3I Atlas is chemically unusual. What we do not know is why. And before you think Earth is the only target worth considering, let me ask you this. What if 3I Atlas is not aiming at us at all? Part 4. The Europa question. Why 3I Atlas might not be aiming at Earth at all. Earth is not the most interesting place in the solar system if you are trying to seed life. Let me explain why an interstellar carrier, if that is what this is, might be more interested in Europa, or Enceladus, or one of the other icy moons with subsurface oceans, than it is in our pale blue dot. Europa, Jupiter's fourth largest moon, has a shell of water ice covering a global ocean that is twice the volume of all of Earth's oceans combined. That ocean is kept liquid by tidal heating, the gravitational squeeze Jupiter exerts on Europa as it orbits. The surface is frozen and radiation blasted, but beneath that ice there is liquid water, dissolved minerals, and possibly hydrothermal vents. If you wanted to plant the seeds of life somewhere protected, somewhere with energy sources and chemical gradients, Europa is a far better candidate than Earth's surface, which is already crowded with established ecosystems. An icy moon offers a blank slate, a place where introduced organisms would not have to compete with billions of years of evolutionary incumbents. Orbital mechanics makes this more plausible than you might think. 3I Atlas is on a hyperbolic trajectory that will take it past Earth and then onward through the solar system. If its velocity and direction are right, and if it can make small adjustments, it could continue toward the Jupiter system, with relatively low delta V requirements. Delta V is the change in velocity needed to alter a trajectory, and in space, delta V is currency. The less you need, the more feasible the mission. A gravitational assist from Earth or Mars could provide some of that delta V for free, nudging the object toward Jupiter without requiring additional propellant. This is speculative, but it is not physically absurd. Missions to the outer solar system use gravitational assists all the time. Methanol's role in subsurface ocean chemistry is particularly interesting. Under Europa's conditions, high pressure, low temperature, radiation flux from Jupiter's magnetosphere, methanol could act as both a carbon source and an energy source for chemosynthetic ecosystems. Chemosynthesis is the process by which organisms derive energy from chemical reactions rather than sunlight. On Earth, deep-sea hydrothermal vents support entire ecosystems based on chemosynthesis, and similar processes could theoretically occur in Europa's ocean. Methanol, when oxidized or reduced in the presence of minerals and radiation, can release energy and produce more complex organic molecules. It is a starter fuel for biochemistry in dark, isolated environments. Intentional targeting versus random pass. The probability of an interstellar object randomly intersecting Jupiter's gravitational sphere of influence is low, but not zero. We have seen 2 confirmed interstellar visitors now, Oumuamua and 2I/Borisov, and statistically, we expect more. But 3I Atlas's chemistry and venting behaviour do not look entirely random. The directional asymmetries, the unusual molecular ratios, the steep power law scaling, these are features that make you wonder whether the object is following a programmed sequence rather than passively responding to solar heating. If it is programmed, then targeting becomes a possibility. If it is natural, then any intersection with Europa would be coincidental. The bioassessment idea is purely speculative, but let me lay it out anyway. December 19th, Earth's closest approach could be a scan, a way for the object, or whatever mechanism controls it, to test whether Earth is biochemically hospitable before committing resources to a deeper insertion. What signals would an advanced probe look for? atmospheric composition. Oxygen and methane together are a strong biosignature because they react with each other and should not coexist unless something is continuously replenishing them. Electromagnetic, anomalies, radio transmissions, modulated signals, anything that suggests technological civilization. Surface temperature and albedo patterns, indicators of liquid water and seasonal cycles. If the object had sensors, and if it were capable of processing that data, it could make a go-no-go decision about whether to adjust its trajectory toward a secondary target like Europa. Counterpoints exist, and they are substantial. Targeting an ice moon requires precision. You need to know where the moon will be in its orbit at the time of your arrival, and you need to adjust your velocity vector to match. That requires propulsion, navigation, and some form of onboard computation or pre-programming. The energy requirements are not trivial. Even a small course correction at interstellar distances requires significant delta V, and we have not seen evidence of large-scale propulsion events. The object is venting molecules, not firing rockets, at least not in any way we have confirmed. Observational tests could settle this. If 3i Atlas changes its venting pattern after planetary flybys, that would be significant. If we detect fragments or secondary objects separating from the main body and trending toward Jupiter, that would be extraordinary. Spectroscopic follow-ups in the coming months, wide-field imaging surveys tracking the object's trajectory, these are the methods we should be using to monitor for anomalies. If the object is natural, it will continue on its hyperbolic path and fade from view. If it is something else, it will give us more clues. But there is another possibility, one that is both more mundane and more disturbing. What if hydrogen cyanide is not a weapon or a prebiotic feedstock? What if it is just exhaust? Part 5. The Serial Killer Interpretation. Revisited. Could HCN be a propellant byproduct? What if cyanide is not a weapon but exhaust? Let me be very clear up front. This is speculative, and it invokes chemistry that is unusual, possibly exotic, and certainly not something we have direct evidence for. But the question is worth asking because it reframes the entire narrative. Hydrogen cyanide can be produced in high temperature, oxidative, or catalytic environments. If you burn hydrocarbons or amines in the presence of nitrogen, you can get nitriles, including hydrogen cyanide, as byproducts. Certain hypothetical thermochemical thrusters, engines that rely on chemical reactions to generate thrust, might produce exactly this kind of exhaust. Imagine an engine that uses organic fuels, carbon and nitrogen-rich compounds, and oxidizes them in a controlled burn. The primary product is carbon dioxide and water vapor. The secondary products could include hydrogen cyanide, ammonia, and other nitrogen-bearing molecules. If 3i Atlas had an engine like that, and if it were using it to make small trajectory corrections, the hydrogen cyanide we are seeing might not be cargo or payload. It might be the leftover smoke from the engine. Alma found that hydrogen cyanide is depleted on the sunward side of the object. If hydrogen cyanide was sublimating passively from the surface, you would expect more of it on the sunward side where the heat is strongest. But we see the opposite. It is stronger on the anti-sunward side, or at least stronger in certain directions that do not correlate with solar illumination. That pattern makes sense if the cyanide is being vented from specific locations like thruster ports, rather than evaporating uniformly from the surface. Shielding could also play a role. If the active regions are on the shaded side of the nucleus, solar heating would be minimal there, and the venting could be driven by internal processes rather than external radiation. Natural analogues exist, but they are not perfect matches. Some solar system comets, like Comet 67P Churyumov-Gerasimenko, show asymmetric jets tied to specific geological features. Cliffs, pits, and fractures can focus volatile release into narrow beams, but 3i Atlas's asymmetry is more pronounced, and the molecular composition is more unusual. Comparing ALMA maps of 3i Atlas with maps of other comets shows differences in both the spatial distribution and the intensity of the emissions. The jets on 3i Atlas seem more directional, more organised, and that organisation is what raises eyebrows. Here is the engineering thought experiment. A vehicle shedding low-mass molecular exhaust to adjust attitude, its orientation in space, while simultaneously carrying a separate reservoir of organic cargo. The exhaust would be optimised for momentum transfer, which means high velocity, low molecular weight particles. Hydrogen cyanide fits that profile. It is light, it is easy to accelerate, and it carries kinetic energy efficiently. The cargo, methanol, would be stored in a different compartment and released separately, possibly from different vents or through a different mechanism. This is how you would design a dual-purpose mission. Propulsion and payload delivery in one package. The feasibility issues are significant. Mass budgets, the amount of fuel required versus the mass of cargo, reaction energetics, the energy released per molecule of fuel burned. These are all constraints that would limit what you could do with a chemically propelled interstellar vehicle. But they are not insurmountable, especially if the technology level is higher than ours. Signatures of propulsive behaviour would show up in the data if they existed. Non-Keplerian accelerations, deviations from the trajectory you would expect based on gravity alone, are one signature. Anomalous light curve periodicities, brightness variations that do not match the object's rotation rate, are another. Recurring jet patterns, venting events that happen at regular intervals or in response to specific conditions would also be telling. So far, we do not have confirmed reports of any of these. The object appears to be following a standard hyperbolic orbit, and its brightness variations seem consistent with a tumbling, irregularly shaped body. But absence of evidence is not evidence of absence. We have not been monitoring 3i Atlas continuously, and subtle accelerations might not show up in the limited data we have. If hydrogen cyanide is exhaust and methanol is cargo, then we are looking at a machine. Or we are looking at nature doing such a convincing impression of a machine that we cannot tell the difference. That ambiguity, that uncertainty is where the real tension lives. Science demands skepticism, but it also demands openness to possibilities that seem absurd until they are not. Oumuamua taught us that. 2i/Borisov taught us that. And now 3i Atlas is teaching us that the line between natural and artificial, between chemistry and engineering, is thinner than we thought. So here is the cliffhanger. If hydrogen cyanide is exhaust and methanol is cargo, who built the ship? Or is nature just that good at masquerading as technology? We are about to go deeper. The next act takes us beyond the payload, into the hidden implications, the statistical anomalies, and the larger mysteries in astronomy that 3i Atlas connects to. Because this is not just about one object. It is about what that object means for everything else we thought we understood. Part 6. The Methanol Power Law. Mystery. Why this scaling shouldn't exist in a natural object. ALMA reports that methanol production from 3i Atlas scales roughly as R sub h to the negative 5.2 power. Let me translate that. R sub h is the heliocentric distance, the distance from the Sun. As 3i Atlas approached the Sun, its methanol output increased dramatically, much more dramatically than normal comets. And that exponent, negative 5.2, is a number that should not exist in a natural sublimating body. Power laws describe how one quantity changes in relation to another. In comet science, we use power laws to describe how production rates of volatiles, molecules like water, carbon monoxide, methanol, change as the comet gets closer to the Sun and heats up. Think of it like an oven. The closer you get to the heat source, the faster ice melts. But the relationship is not linear. Doubling the temperature does not double the sublimation rate. It increases it exponentially, or in this case, according to a power law. For typical comets, the exponent ranges from about -2 to -4. Water ice usually clocks in around -3. Carbon monoxide, which sublimates at lower temperatures, might be closer to -2. Methanol, being intermediate, should fall somewhere in that range. But 3i Atlas gives us -5.2. That is steeper than anything we normally see, and it suggests something other than simple ice melting is driving the methanol release. Let me walk you through 3 natural models and why each one struggles. Pure sublimation. If methanol ice was sitting on the surface, absorbing sunlight and turning directly from solid to gas, you would expect an exponent closer to -3 or -4. The steeper exponent suggests that heating alone is not enough. Devolatilization of clathrates. Clathrates are ice structures where one molecule, like water, forms a cage around another molecule, like methanol. When the clathrate breaks apart, it releases the trapped gas in a burst. But even clathrate decomposition typically produces exponents in the -3 to -4 range. You would need very specific temperature thresholds and a very large reservoir of clathrates to push it to -5.2. Thermal cracking of organics. If complex organic molecules on the surface are breaking down into smaller fragments as they heat up, that could release methanol as a byproduct. but thermal cracking is usually a gradual process, not a runaway one, and the exponent should reflect that. So here is the runaway catalysis hypothesis, and this is where things get interesting. Imagine a surface catalyst, a mineral or a microstructure, that once heated by sunlight triggers exothermic reactions. Exothermic means the reactions release heat. That released heat further accelerates the reactions, which release more heat, creating a feedback loop. Methanol production goes into overdrive, not because the sun is heating the ice faster, but because the ice is heating itself through chemical reactions. If you have ever watched a pile of damp hay spontaneously combust, you have seen this principle in action. Decomposition releases heat, heat accelerates decomposition, and suddenly you have a fire that was not directly ignited. On 3I Atlas, the feedback could be chemical rather than combustive, but the mathematics would be similar. Runaway systems produce steep power laws because small changes in temperature can trigger large changes in reaction rates. Laboratory analogues exist, sort of. When you irradiate organic ices with ultraviolet light or energetic particles, you can trigger secondary chemistry. Methanol can form from carbon monoxide and hydrogen under irradiation. Formaldehyde can decompose into methanol and carbon dioxide under certain conditions. These reactions show nonlinear temperature dependence in the lab, meaning their rates do not increase smoothly with temperature. They can have thresholds, activation energies that once crossed, cause production rates to spike. If 3I Atlas has a surface rich in such reactive materials, and if those materials are being activated as the object approaches the sun, you could get the steep power law we're seeing. Observational tests that would support runaway catalysis include time-dependent spectral changes. If the methanol lines got stronger or weaker abruptly, rather than smoothly, that would suggest threshold behavior. Abrupt spikes in production rate, sudden increases that do not correlate with steady solar heating, would also be evidence. Correlated thermal emission, if infrared observations showed hotspots on the surface that coincided with methanol venting, that would support the exothermic reaction hypothesis. ALMA has the spectral resolution to detect some of these features, but whether it saw them in the limited observation windows is unclear from the publicly available data. We need more detailed analysis of the time series to know for sure. An alternative engineered scenario involves an internal reagent bank. Imagine containers or reservoirs inside the object that are sealed until triggered by heat or sunlight. Once the seals break or the valves open, reagents mix, react and produce methanol in a controlled or semi-controlled release. The steep power law could reflect the opening sequence of those reservoirs, with more of them activating as the object gets closer to the sun. This sounds like science fiction, and maybe it is, but the energetics and mass requirements are not prohibitive. A few tons of reactive chemicals could produce the methanol output ALMA measured. The question is whether nature can build such a system, or whether it requires deliberate engineering. The implications are profound either way. If this is natural, we have discovered a new class of reactive comet, a type of object that undergoes catalytic chemistry on its surface and produces volatile surges that standard models cannot predict. That is a major finding in planetary science. If this is engineered, if the object demonstrates controlled release mechanisms, then we are looking at technology, and that would be the biggest discovery in human history. Both conclusions rest on the same data point, negative 5.2, a number that should not exist but does. Next, we're going to zoom out and ask where an object like this could have come from, and whether its chemistry tells us anything about the stellar nursery that birthed it. Part 7. The Interstellar Disk. Hypothesis. Did 3I Atlas come from a methanol-heavy dead system? Protoplanetary disks, the swirling clouds of gas and dust that surround young stars and eventually form planets, vary wildly in composition. Some are carbon-rich, some are oxygen-rich, some have unusual abundances of nitrogen, sulfur or metals, and some, a rare subset, are methanol-heavy. If 3I Atlas formed in a methanol-heavy disk, that would explain its chemistry without invoking anything exotic or engineered. Let me show you how. Methanol forms in interstellar space and in protoplanetary disks through surface chemistry on dust grains. Carbon monoxide, one of the most abundant molecules in space, lands on a cold grain, gets hydrogenated by hydrogen atoms also landing on the grain, and becomes methanol. This process happens over millions of years in the dense cold regions of molecular clouds. When a protoplanetary disk forms, it inherits the chemistry of the cloud it collapsed from. If that cloud was particularly rich in carbon monoxide and hydrogen, the disk would be methanol-rich. Planetesimals, the building blocks of planets and comets that formed in such a disk, would incorporate that methanol into their ice. C/2016 R2 is our closest analogue, and it is worth examining. This comet, observed in 2017 and 2018, had an unusually high abundance of carbon monoxide and nitrogen, along with elevated methanol. Its composition suggested it formed in a colder, more carbon-rich environment than typical solar system comets. The parallels to 3I Atlas are not perfect, but they are suggestive. If R2, which is from our solar system, can have such unusual chemistry, then an interstellar visitor from a different system could have even more extreme ratios. Here is a plausible origin story. In another star system, a few light years or a few hundred light years away, a protoplanetary disk rich in methanol and other organics formed planets and moons. One of those moons, analogous to our Kuiper Belt objects, was an icy body with a methanol-rich veneer. Gravitational interactions, either with a migrating gas giant or with another star passing nearby, ejected that icy body from its home system. It spent millions or billions of years drifting through interstellar space, slowly accumulating damage from cosmic rays and interstellar dust impacts, until it entered our solar system on a hyperbolic trajectory. That body is 3I Atlas. Its chemistry is a fossil record of its birth environment, a snapshot of a disk we will never directly observe. Isotopic signatures would settle this question decisively. Isotopes are versions of the same element with different numbers of neutrons, deuterium to hydrogen ratios, carbon-13 to carbon-12 ratios, nitrogen-15 to nitrogen-14 ratios. These are all diagnostic of the temperature and environment where a molecule formed. If 3I Atlas has deuterium enrichment, that would suggest it formed in a very cold environment, possibly in the outer regions of a protoplanetary disk, or even in the interstellar medium itself. If its carbon isotope ratios are different from solar system values, that would prove it formed around a different star. ALMA can measure some isotopologues, molecules with different isotopic compositions, but doing so requires long integration times and specific observing windows. Future millimetre and submillimetre observations should prioritise isotopic measurements. They are the fingerprints that will tell us where this thing came from. The consequences of a methanol-rich origin go beyond 3I Atlas. If interstellar chemistry is diverse enough to produce objects with such extreme compositions, then the seeding of prebiotic molecules across the galaxy may be more common than we thought. Every star system could be receiving a slow rain of organic-rich fragments from other systems. Panspermia, the idea that life spreads through space on comets and asteroids, does not require intent if the raw materials are being distributed naturally. You do not need a gardener if the garden seeds itself. Objections exist. Methanol-heavy objects might be rare, even in methanol-rich disks. Selection effects could bias our observations. We detect the objects that are bright, active and close, not necessarily the objects that are typical. 3I Atlas might be an outlier, a 1 in a billion composition that does not represent the average interstellar population. We simply do not have enough interstellar visitors yet to know what typical looks like. What to watch for? JWST, the James Webb Space Telescope, has the sensitivity and spectral resolution to detect isotopologues that ALMA cannot. Long baseline ALMA mapping, using the array's full resolution, could reveal finer details of the spatial distribution and kinematics of the molecules, and any retrieval or intercept mission concept, a probe sent to rendezvous with 3I Atlas before it leaves the solar system, would give us direct samples. That is the gold standard. Until we have samples, we are making educated guesses based on remote sensing. But while we are guessing, let me ask you something truly strange. What if cyanide is not poison or exhaust? What if it is an anesthetic? Part 8. The opioid sedation. Theory. Cyanide as a planetary anesthetic. Could cyanide be a planetary anesthetic? I am labelling this up front as fringe, speculative and possibly absurd, but the idea is compelling enough that it deserves examination, if only to understand why it fails. Hydrogen cyanide at low concentrations does not just kill, it alters biochemistry. In mammals, low-dose cyanide exposure affects neurotransmitter systems. It interferes with dopamine metabolism, alters serotonin pathways, and can produce effects ranging from euphoria to stupor depending on dose and duration. In microbes, cyanide can inhibit certain metabolic pathways without causing cell death. The organism slows down, becomes less competitive, less aggressive in resource acquisition. This is not sedation in the human sense, but it is metabolic suppression, and the functional result is similar. An organism exposed to low-dose cyanide is easier to outcompete. Here is the speculative mechanism. Low-dose hydrogen cyanide acts to suppress aggressive or competitive actors in a biosphere, clearing ecological niches for colonizing microbes. If you were seeding a planet with your own organisms, you would want to give them an advantage. One way to do that is to suppress the existing biology, not kill it outright, but slow it down, make it less able to defend its territory. Methanol comes in as the feedstock for your colonizers, the methylotrophs that can metabolize alcohol and thrive in environments where other organisms are struggling. It is a 1-2 punch, but surgical, targeted, designed to shift the equilibrium rather than wipe the board clean. Plausibility requires calculation. What atmospheric column concentrations of hydrogen cyanide would be needed to affect microbes? Let me give you rough numbers. To produce measurable metabolic suppression in bacteria, you need concentrations in the parts per million range in their immediate environment. In Earth's atmosphere, that would require deposition rates far higher than what ALMA measurements suggest 3I Atlas is releasing. To affect mammals, you need even higher concentrations and over larger areas. Whole Earth sedation, the idea that cyanide from 3I Atlas could drug the entire planet, is numerically implausible. The dilution factors are too extreme, and the atmospheric mixing times are too fast. By the time any cyanide reached Earth's surface, it would be so dispersed that it would be undetectable. But, and this is where it gets interesting, magnetospheric and ionospheric interactions could concentrate particles in specific latitudes or altitudes. Earth's magnetic field funnels charge particles toward the poles, creating the auroras. If hydrogen cyanide molecules or cyanide-bearing dust grains were ionized, they could follow similar paths. You could get localized enhancements in the upper atmosphere at high latitudes, regions where the concentration might be orders of magnitude higher than the global average. Whether that concentration would be high enough to affect biology is still questionable, but it is less implausible than whole planet exposure. This connects to historical and ethical thought experiments about chemical agents. Humans have used sedatives, paralytics, and chemical incapacitants in warfare and agriculture. The idea of suppressing a population before colonization is not alien to our own behavior. Repeating those analogies in a cosmic context is ethically charged, and I want to be clear that I am not advocating for this interpretation. I am presenting it as a narrative possibility, a way to think about the chemistry we're seeing, not as a factual claim. Epistemic reminder. This is interesting for storytelling. It is weak for hard science. There is no direct evidence of atmospheric transport, no measurements of cyanide concentrations in Earth's upper atmosphere that correlate with 3I Atlas's approach, and no biological or ecological signals that would support this hypothesis. It is a thought experiment, and it should remain one unless new data forces us to revisit it. Next, we're going to take the gardener metaphor seriously and ask what a terraforming cocktail would actually look like if someone were trying to rewrite a biosphere from scratch. Part 9. The Terraforming Cocktail, Methanol. Cyanide as a biosphere DNA rewrite primer. What if the gardener is not planting us, but planting something tailored to a different biochemistry? Let me be very clear. This is speculative framing, a narrative device, not a factual assertion. But the chemistry of 3I Atlas, methanol plus cyanide, reads like a recipe for ecosystem engineering if you squint hard enough. Methanol supports methylotrophs, bacteria that use methanol as their primary carbon and energy source. These organisms are versatile, resilient, and can thrive in environments where sugars and other carbon sources are scarce. On Earth, methylotrophs are found in soil, freshwater, marine environments, and even in extreme habitats like hydrothermal vents. If you wanted to introduce a metabolism that could establish itself quickly and survive in a wide range of conditions, methylotrophy is a good choice. Cyanide, meanwhile, can selectively suppress organisms that lack cyanide resistance pathways. Many bacteria and archaea have enzymes that detoxify cyanide, converting it into less harmful compounds. Organisms without those enzymes are at a disadvantage, so if you release methanol and cyanide together, you create a selective pressure. Methylotrophs with cyanide resistance flourish. Everything else struggles. Here is a conceptual ecosystem engineering diagram described in words. Input is methanol delivered by 3i Atlas. Primary colonizers are methylotrophic bacteria with cyanide resistance. These organisms establish biofilms, consume the methanol, and begin altering the local chemistry. Ecosystem changes include shifts in pH, oxygen production or consumption, accumulation of organic byproducts. Over time, the colonizers modify the environment to suit their metabolism, creating niches for secondary organisms that depend on the byproducts of methylotrophy. The end result is an ecosystem that looks different from the one that existed before, not because it was destroyed, but because it was outcompeted and reshaped. The steps an engineered terraforming cocktail would need are specific and non-trivial. Selective suppression. You need an agent that targets existing biology without wiping it out entirely. Cyanide, in the right dose, fits that role. Nutrient feedstock. You need a carbon and energy source for your colonizers. Methanol fits that role. Protective microenvironments. Your colonizers need places to establish themselves where they are shielded from radiation, desiccation, or predation. Dust grains, ice particles, or organic polymers could provide those niches. Delivery timing. The suppression agent and the feedstock need to arrive together, or in rapid succession, to maximize the advantage of the colonizers. If the cyanide arrives too early, it dissipates before the methanol shows up. If the methanol arrives too early, it gets consumed by existing organisms before the cyanide suppresses them. Timing is everything. Technical hurdles are substantial. Dose control is nearly impossible at interstellar distances. You cannot fine-tune the concentration of molecules released from a comet to match the requirements of a specific biosphere. Persistence is another issue. Methanol and cyanide are both reactive and will degrade or disperse over time. Bystander effects, unintended consequences for non-target organisms, are unavoidable. Ecological systems are complex, and introducing new selective pressures always produces outcomes you did not predict. Historical context matters here. Directed panspermia, the idea that life on Earth was intentionally seeded by an advanced civilization, was proposed in the mid-20th century by scientists like Francis Crick and Leslie Orgel. They were serious researchers asking whether the origin of life on Earth could have an extraterrestrial explanation. Modern synthetic biology debates on planetary protection ask similar questions. If we send microbes to Mars either intentionally or accidentally, are we terraforming it? Are we contaminating it? The ethics and policy around these questions are still being developed, and 3i Atlas forces us to think about them in reverse. What if someone else is seeding us? Plausible timeline if such seeding were attempted. Immediate chemical effects, measurable within days or weeks of exposure. Microbial colonization, establishment of biofilms and communities, would take years to centuries depending on environmental conditions. Ecosystem shift, visible changes in species composition and function, would take longer, possibly millennia. Nothing like this has been observed yet. There are no reports of sudden microbial blooms, no shifts in atmospheric trace gases, no ecological anomalies that correlate with 3i Atlas's approach. So if this is terraforming, it is either in the very early stages or it has not started yet. Detection signals to watch for include sudden shifts in atmospheric methane, carbon dioxide or other trace gases, emergence of methylotrophic biosignatures, isotopic ratios in organic matter that suggest methanol metabolism, changes in soil or marine microbiology, increases in cyanide-resistant bacteria or methylotrophs in specific regions. Observational strategies would involve atmospheric monitoring, soil and water sampling, and long-term ecological surveys. None of this is happening in a coordinated way right now, and maybe it should be. Sober assessment, spectacular to imagine, extraordinarily difficult to execute and detect. Use this as a narrative device, not a factual claim. The chemistry is suggestive, but suggestion is not proof, and proof is what science requires. Before we leave Act 2, there is one more question worth asking. What if 3i Atlas is not delivering a payload at all? What if it is testing us? Part 10, the 3i Atlas Intelligence Test. Is the object probing before acting? Rather than delivering a payload, what if 3i Atlas is probing planetary environments to assess suitability? This would be a low-risk reconnaissance, a way to gather data before committing to a more invasive action. A scientific probe designed to evaluate. A biosphere would measure several things remotely. Atmospheric composition is first. Oxygen, methane and ozone are key. Oxygen and methane together are a strong biosignature because they react and should not coexist in equilibrium unless something is replenishing them. Ozone forms from oxygen and indicates photosynthesis. Oxidation states, the balance between reduced and oxidized compounds, tell you about the chemical energy available in the environment. Seasonal variability matters because it indicates a stable climate with predictable cycles. Magnetic signatures are important because a magnetic field protects the surface from radiation, making it more hospitable for biology. A probe measuring all of these could build a profile of habitability and decide whether to proceed with seeding or to move on to another target. ALMA evidence of compositional changes over the observation window is present in the data, but interpreting it is tricky. 3i Atlas showed time-variable production rates for methanol and hydrogen cyanide, but those variations could be explained by rotational modulation, the object spinning and exposing different active regions to sunlight at different times. They could also be explained by thermal cycling, regions heating up and cooling down as the object orbits. Whether any of that constitutes probing behaviour, intentional modulation of venting in response to external stimuli is not clear from ALMA alone. The data show variability, but variability is not the same as intent. Telemetry proxies we can search for include targeted venting when close to certain planets. If 3i Atlas increased its methanol output as it approached Earth and decreased it afterward, that would be interesting. Jet pulsing correlated with planetary alignments, venting events that happen when the object is at specific angles relative to Earth or Jupiter, would also be suggestive. Non-random orientation of active regions, if the jets consistently point toward or away from specific targets, that would imply some form of targeting or awareness. Counter-explanations are abundant. Natural thermal cycling can produce periodic variations. Rotational modulation of jets is common in comets and does not require intelligence. Impact-driven exposures, where small meteoroid impacts fracture the surface and release buried volatiles, can create sudden bursts of activity. Discriminating between these natural processes and intentional behaviour requires careful analysis. Frequency content analysis of jet periodicity versus rotation is one method. If the jets pulse at frequencies that do not match the rotation period, that is anomalous. Polarimetry for dust alignment could reveal whether particles are being organised by magnetic fields or other directional forces. Coordinated solar wind monitors, satellites measuring the interplanetary magnetic field and solar wind speed, could show whether plasma conditions correlate with venting events. Here is a thought. Experiment. How would you design an observational campaign to test for intentional probing using existing telescopes? Step 1. Spectral cadence. Observe 3i Atlas as frequently as possible with high-resolution spectrographs. Look for time-dependent changes in emission lines that do not correlate with solar distance or rotation. Step 2. Polarimetry. Measure the polarization of scattered light from the dust coma. Organized or aligned particles produce polarization signatures that random dust does not. Step 3. Coordinated monitoring. Combine optical, radio and plasma measurements. If venting events coincide with specific magnetic field configurations or solar wind conditions, that could indicate the object is responding to its environment. Narrative cliffhanger. Whether 3i Atlas is natural, engineered or a hybrid, the scientific and societal stakes are enormous. And this is not the only weird thing happening in astronomy this year. The wider cosmic landscape is shifting, and 3i Atlas is part of a larger pattern. Let me show you what else is out there. Part 11. December. AI model. Breakthrough. Neural nets predict stellar chemistry from light curves alone. In December of 2025, a neural network model achieved something that should make you rethink what intelligence means, artificial or otherwise. It can infer stellar abundances, the chemical composition of a star from limited photometry, the brightness variations you see when you point a telescope at it. No spectroscopy required, just light curves, the ups and downs of brightness over time, and a trained algorithm that has learned to read chemistry in the flicker. This matters because it changes what is possible with minimal data. Traditionally, if you wanted to know what a star is made of, you needed spectroscopy. You needed to split the starlight into its component wavelengths and look for absorption lines, the dark gaps in the spectrum where specific elements absorb specific colours. That requires specialised instruments, long integration times and clear skies. Photometry, on the other hand, is cheap and fast. Any telescope with a camera can measure brightness. The fact that you can now extract chemical information from brightness alone means you can analyse thousands of stars in the time it used to take to analyse one. Implications for advanced civilizations are immediate. If a self-evolving probe or an intelligent artifact wanted to assess planetary habitability remotely, it would not need to get close. It would not need to deploy spectrographs or send back data. It could analyse starlight, infer the composition of the stellar system, and make educated guesses about which planets might be worth investigating. A star rich in heavy elements is more likely to have rocky planets. A star with unusual carbon to oxygen ratios might produce planets with exotic chemistries. A neural network running on minimal hardware could process this data in real time and make go-no-go decisions about where to direct attention or resources. Connection to 3i Atlas. If the object carried a predictive algorithm, or if it had onboard sensors that could measure starlight and derive chemistry, it could have been assessing our solar system long before it arrived. It could have known, based on the sun's spectrum, that Earth is here, that Earth has oxygen and water, and that Earth is a candidate for seeding or observation. This is speculative but plausible. The technology exists now in our labs, in our universities. It is not a stretch to imagine it deployed on an interstellar mission. Simple demonstrations of the concept are visually compelling. You can show a light curve, a jagged line of brightness over time, and overlay it with the inferred chemical abundances: iron, magnesium, silicon, carbon, oxygen, all extracted from the pattern of flickers. The neural network does not understand chemistry the way a human does. It has learned correlations, patterns in the training data, that link brightness variations to elemental abundances. But the output is accurate enough to compete with traditional spectroscopy in many cases. That is both impressive and unsettling. Methodological limits are real. False positives happen when the light curve resembles another star with different chemistry. Dependence on training data means the network only works well for stars similar to those it was trained on. Biases in the training set propagate to the predictions. Human confirmation is necessary, especially for unusual or extreme cases, but the speed and scalability of the method make it a powerful tool for large surveys, and that power could be leveraged by any intelligence, natural or artificial, trying to map habitable zones across the galaxy. Observational countermeasures. What could telescopes detect if 3i Atlas carried data processing signatures? Directed radio emissions, narrow band signals that might indicate telemetry or communication are one possibility. Thermal anomalies, heat signatures that do not match passive solar heating, could indicate onboard electronics or reactors. Modulated optical signals, brightness variations that encode information rather than reflecting natural processes, would be a smoking gun. None of these have been detected, but we have not been looking for them systematically. If you do not search, you will not find. Modern AI blurs the line between natural emergent behaviour and engineered information processing. A neural network trained on stellar spectra is not alive, not conscious, but it performs a function that looks intelligent. If we can build such systems, why assume nature cannot? And if nature can, how do we distinguish between a comet with reactive chemistry and a probe with onboard computation? The answer is we might not be able to, not without close inspection. Next, we are going to talk about space-time itself, and why recent constraints on quantum gravity suggest that the universe might be smoother, more coherent than we thought. That smoothness has implications for long-range transport of matter and information, and it ties directly into the question of how 3i Atlas could have travelled so far without falling apart. Part 12, the 2025 Quantum Gravity Constraint Update. Space-time foam isn't behaving as expected. Space-time, at the smallest scales, is supposed to be foamy. Quantum gravity theories predict that if you zoom in far enough, down to the Planck length, 10 to the negative 35 meters, space-time stops being smooth. It writhes, fluctuates, bubbles with quantum uncertainty. That foam should have observable consequences, tiny distortions in the paths of photons travelling across cosmic distances. But the latest observational constraints from 2025 tighten the limits on how foamy space-time can be, and the answer is less foamy than expected, smoother, more coherent. What this means in plain language is that space-time fluctuations, the noise at the quantum level, are smaller than some models predicted. Photons travelling from distant quasars or gamma-ray bursts show less blurring, less distortion, than they should if space-time were maximally turbulent. The implications are subtle but profound. A smoother space-time allows for better long-range coherence. Signals, particles, even macroscopic objects can travel farther without degrading or scattering. It is like the difference between sailing on a calm sea versus a choppy ocean. The calm sea lets you maintain course and speed with less effort. Relevance to interstellar carriers is speculative but intriguing. If space-time is smoother than expected, then long-range coherence of particle or filament travel could be higher. A stream of nanograins, a plume of molecules, even a macroscopic object like 3I Atlas could traverse interstellar space without as much disruption from quantum fluctuations. That does not make interstellar travel easy, but it makes it slightly less impossible. And when you're dealing with timescales of millions of years, slightly less impossible might be enough. Observational evidence comes from high-energy astrophysics. Gamma-ray bursts, the most energetic explosions in the universe, emit photons across a wide range of energies. If space-time foam were significant, high-energy photons would arrive at Earth slightly delayed relative to low-energy photons, because they interact more strongly with the foam. Observations show that the delays are smaller than some quantum gravity models predict, which means the foam, if it exists, is less pronounced than those models suggest. The limits are still being refined, and different models make different predictions, so this is an ongoing area of research. Connection to 3I Atlas. If the object or its wake is exploiting low-noise corridors, regions of space-time where fluctuations are minimal, it could guide particulate flows more reliably than random dispersion would allow. Plasma filaments, streams of charged particles, could follow magnetic field lines with less scattering. Dust grains could maintain coherent trajectories over astronomical distances. This does not require technology. Natural processes could take advantage of space-time smoothness. But it opens the door to engineered solutions as well. Analogies help. Imagine sending a message across a noisy room. If the room is quiet, your message arrives clearly. If the room is loud, your message gets garbled. Space-time smoothness is the quietness of the cosmic room. A quieter room means clearer signals, more reliable transport, better preservation of structure over distance. For an interstellar object carrying chemicals or particles, that clarity could mean the difference between arriving intact and dispersing into irrelevance. Speculative scenarios. Technology or natural processes using coherent plasma filaments to guide particulate flows. Magnetic field lines threading through smoother space-time could act as rails, channels that keep particles on track. We see hints of this in solar wind structures and in the interstellar medium. Whether it is happening with 3I Atlas is unknown, but the physics allows for it. Next, we're going to look at radio structures near a pulsar wind nebula that show ordering you would not expect from random plasma turbulence. Ordered structures imply mechanisms, and mechanisms imply possibilities for long-distance transport that we have not fully considered. Part 13. Strange radio structures detected near a pulsar wind nebula. Interferometric observations in 2025 revealed filamentary radio structures near a pulsar wind nebula that are more ordered than they should be. Pulsars are rapidly spinning neutron stars that emit beams of radiation and drive winds of relativistic particles. Those winds collide with the surrounding interstellar medium and create nebulae, glowing clouds of plasma and magnetic fields. Normally, the structures in those nebulae are turbulent, chaotic, driven by instabilities and shocks. But the new observations show filaments, radial structures, that are aligned, organised, almost geometric. When I say geometric, I mean the kind of regularity you would expect from a designed system, not from random astrophysical chaos. The filaments run parallel to each other in some regions, radiate outward from central points in others, and maintain consistent spacing over distances of light years. That should not happen in turbulent plasma, and yet there it is, captured in high-resolution radio maps from interferometers spanning continents. Let me give you context on what pulsars actually do, because the physics here is extreme. A pulsar is the collapsed core of a massive star that exploded as a supernova. What remains is a ball of neutrons roughly 20 kilometers across but containing 1.4 to 2 solar masses. That density is incomprehensible. A teaspoon of neutron star material would weigh 1 billion tons on Earth. The pulsar spins, sometimes hundreds of times per second, and as it spins, its magnetic field, which is trillions of times stronger than Earth's, sweeps through space. Charged particles get caught in that field, accelerated to nearly the speed of light, and ejected as a wind. That wind is not gentle. It carries energy equivalent to thousands of suns, concentrated into narrow jets and diffuse flows that slam into the surrounding gas at relativistic speeds. When that wind hits the interstellar medium, the gas and dust that fills the space between stars, the collision creates a shock front. The shock heats the gas to millions of degrees, ionizes it, and creates the nebula, a glowing shell of plasma visible in X-rays, optical light, and radio waves. The most famous example is the Crab Nebula, the remnant of a supernova observed by Chinese astronomers in 1054 AD. The Crab has a pulsar at its centre, spinning 30 times per second, and the entire nebula is powered by the energy that pulsar injects. The structures inside the Crab are messy, filamentary, yes, but chaotic, driven by turbulence and magnetic instabilities. The new observations from a different pulsar wind nebula show something cleaner, more organised, and that is what has researchers scratching their heads. Plasma physics basics. Plasma is ionised gas, a soup of electrons and ions that responds to electric and magnetic fields. It is the fourth state of matter, distinct from solid, liquid, and gas, and it behaves in ways that seem almost alive. Under the right conditions, plasma can self-organise. Magnetic fields can thread through it, creating channels and filaments that persist for long periods. This is not magic. It is magnetohydrodynamics, the study of how conducting fluids behave in magnetic fields. The equations are complex, non-linear, and notoriously difficult to solve, but the basic principle is simple. Moving plasma generates magnetic fields, and magnetic fields influence how plasma moves. The feedback between the two can create stable structures, patterns that resist dispersion and last far longer than you would expect in a turbulent environment. Examples from the solar system help make this tangible. The Sun's corona, its outer atmosphere, is filled with coronal loops, arcs of plasma that follow magnetic field lines and can persist for days or weeks. The solar wind, the stream of charged particles the Sun constantly emits, is not uniform. It is filamentary, with fast streams and slow streams separated by boundaries that remain coherent over millions of kilometres. Earth's aurora, the northern and southern lights, are visible manifestations of plasma self-organisation. Charged particles from the solar wind funnel along Earth's magnetic field lines, converging at the poles, creating curtains and arcs of light that ripple and dance in patterns governed by magnetohydrodynamic instabilities. All of these are natural, well-understood examples of plasma creating structure. But the degree of organisation seen in these new observations is higher than most models predict for turbulent environments, especially environments as violent as a pulsar wind nebula. The filaments are not just present, they are regular. Spacing between adjacent filaments is consistent, roughly 1/10 of a light year in some regions. Orientation is coherent over distances of several light years, far larger than the characteristic turbulence scale. Width of individual filaments is narrow, often less than 1/100th of a light year, suggesting they are confined by magnetic pressure rather than spreading freely. These properties are unusual. Turbulence tends to create a hierarchy of structures, big eddies spawning smaller eddies, fractal patterns with no preferred scale. What we are seeing here is different. It is organised, almost modular, as if something is imposing order on the chaos. Tie to 3I Atlas. Ordered plasma could form collimated channels that steer nanodust, molecules, or larger particles over astronomical distances. If 3I Atlas is weight couples to the interplanetary magnetic field, and if that field has coherent structure, the weight could follow filamentary paths rather than dispersing uniformly. This is an astrophysical mechanism, not an engineered one, but the functional result is the same. Particles are guided, transported, delivered to locations they would not reach through random diffusion. Let me walk you through how this could work. 3I Atlas vents methanol and hydrogen cyanide, along with dust grains and other particles. Some of those particles are charged, either because they are ionized molecules, or because they are dust grains that have picked up or lost electrons through interactions with ultraviolet light or the solar wind. Charged particles do not move freely through space. They spiral along magnetic field lines, trapped by the Lorentz force, the same force that confines plasma in fusion reactors and creates the auroras on Earth. The interplanetary magnetic field, the magnetic field carried by the solar wind, is not uniform. It has structure, it has filaments, regions where the field is stronger or more organized, and it has boundaries, surfaces where the field direction changes abruptly. If 3I Atlas's wake intersects one of these structures, the charged particles in the wake could be channeled along it, guided like water in a pipe. The filament acts as a highway, a low-resistance path for particles to follow. Uncharged molecules like methanol would not be directly affected, but they could be dragged along by collisions with charged particles, or they could condense onto charged dust grains and hitch a ride. The result is that the wake, instead of dispersing isotropically in all directions, could become anisotropic, concentrated along specific directions defined by the magnetic field geometry. Pros and cons. Plausible plasma self-organization is well documented in laboratory experiments and in space plasmas. Solar wind filaments, coronal loops, and auroral arcs all show ordered structure. We have decades of observations and theoretical work supporting the idea that plasma can organize itself without external intervention. But artificial collimation, deliberate shaping of plasma flows using external fields or engineered structures, is also possible in principle. Humans have built plasma confinement devices, like tokamaks and stellarators, that use carefully designed magnetic fields to guide and control plasma. If a sufficiently advanced civilization wanted to steer interstellar or interplanetary flows, they could, in principle, deploy magnetic structures to do it. The question is how you tell the difference. Discriminating between the two requires diagnostic predictions. Natural self-organization tends to produce fractal structures, repeating patterns at different scales. You see filaments within filaments, a hierarchy that reflects the turbulent cascade from large to small. Artificial collimation might produce more regular periodic structures. Think of the difference between a river delta, which branches chaotically, and a network of irrigation canals, which branches in planned, geometrically regular ways. The pulsar wind nebula observations show something intermediate. Some regions are fractal, others are more regular, and that ambiguity is what makes this interesting. Polarimetry, the measurement of polarized light or radio emission, can reveal the orientation and strength of magnetic fields, which in turn tells you whether the ordering is intrinsic or imposed. If the magnetic field directions in the filaments align with the overall large-scale field of the nebula that suggests self-organization, if they deviate, if they form patterns that do not match the ambient field, that would be anomalous and worth investigating. Observational follow-ups, time series measurements to see whether the filaments evolve, merge, or disappear. Natural structures change. They are dynamic, evolving on timescales set by the flow speed and the size of the structure. For a pulsar wind nebula, that might be years or decades. If the filaments are still there in 10 years, looking the same, that would be interesting. If they are gone, replaced by new structures, that would support the natural turbulence interpretation. Spectroscopy to measure the velocity and temperature of the plasma. Filaments formed by shocks or magnetic reconnection should have specific temperature and velocity signatures. Measuring those can tell you what physical process is responsible. Correlation with particle detectors to see whether the filaments are actually transporting matter or just organizing existing plasma. If the filaments are channels for particle transport, you should see enhanced particle flux along them, detectable by spacecraft equipped with charged particle detectors or dust counters. For 3I Atlas specifically, monitoring the wake with radio telescopes could reveal whether similar structures are forming behind the object. If they are, and if those structures point toward Earth or Jupiter, that would be worth noting. Radio observations can detect ionized gas and measure its density, temperature, and magnetic field. If 3I Atlas's wake shows filamentary structure in radio emission, that would suggest magnetic channeling is happening. If the filaments are oriented toward Earth, that would mean some fraction of the wake is being preferentially directed our way, increasing the probability of chemical delivery despite the large distance. What to watch for? Polarimetry of the wake, looking for aligned dust grains or organized magnetic fields. Dust grains in a magnetic field tend to align with the field direction, and aligned grains produce polarized light when they scatter sunlight. Measuring that polarization tells you the field orientation and strength. Coordinated observations with solar wind monitors to see if plasma conditions correlate with venting events. If 3i Atlas vents more strongly when the solar wind is fast or when the interplanetary magnetic field has a particular orientation, that would suggest the object is responding to its environment, either passively or actively. Wide-field imaging to track the full extent of the wake and see if it forms filaments or remains diffuse. Optical and infrared telescopes can image the scattered light from dust in the wake. If that light shows structure, bright streaks or arcs rather than a uniform glow, that would indicate the wake is not dispersing uniformly. The broader point is that space is not empty, and it is not random. There are structures, channels, pathways that can guide matter and energy over vast distances. The solar wind is structured, the interplanetary magnetic field is structured, the local interstellar medium, the gas and dust in our immediate galactic neighbourhood is structured. We see filaments in hydrogen alpha emission maps, in far infrared dust maps, in radio continuum surveys. The galaxy is threaded with magnetic fields that organise gas and dust into lanes and voids. Stars form in those lanes and planets form from the dust, so the large-scale structure of the magnetic field influences where and how planetary systems develop. If interstellar objects like 3i Atlas are travelling through this structured medium, they are not moving through a featureless void. They are navigating a maze, and the walls of that maze are magnetic. Whether 3i Atlas is using those pathways intentionally or accidentally, the result is the same. Its chemistry can travel farther and more coherently than naive dilution models suggest. Naive models assume isotropic dispersion, particles spreading uniformly in all directions, dilution governed by inverse square law. But if magnetic, channeling is happening, if the wake is being funneled along filaments, then the dilution is anisotropic, weaker in some directions and stronger in others. Earth might be in one of the stronger directions. We will not know until we measure it, and December 19th is our chance to do exactly that. Ground-based radio telescopes, space-based particle detectors, atmospheric chemistry monitors, all of them should be pointing at 3i Atlas and its projected wake path, looking for filaments, looking for structure, looking for signs that the universe has ways of delivering chemistry across distances we thought were prohibitive. Next, we're going to talk about a laboratory breakthrough that makes methanol far more dangerous or far more useful than we thought. Part 14. Breakthrough in Terran synthetic biology. New precursor pathway. Synthesizes RNA bases from simple alcohols. In 2025, a laboratory at a major research university demonstrated a new chemical pathway where methanol, under broadband irradiation, produces RNA precursors. This is not a minor finding. This is a potential revolution in our understanding of how life's building blocks can form under realistic prebiotic conditions. Let me be clear about what makes this revolutionary. For decades, origin of life researchers have struggled with the RNA world hypothesis, the idea that RNA came before DNA and proteins in the evolution of life. The problem has always been explaining how RNA could form spontaneously on early Earth, or anywhere else for that matter, without enzymes to guide the reactions. Ribose, the sugar backbone of RNA, is notoriously difficult to make in prebiotic conditions. It forms in tiny yields, degrades quickly, and competes with dozens of other sugars that nature produces more readily. Nucleobases, the information-carrying parts of RNA, have their own synthesis problems. They require specific conditions, specific starting materials, and they tend to fall apart as fast as they form. The new methanol pathway solves, or at least sidesteps, many of these problems in a way that feels almost too convenient. The chemistry goes like this. Methanol is oxidized to formaldehyde by ultraviolet light or energetic particles. This is step 1, and it is straightforward. Methanol, chemical formula CH3OH, loses hydrogen atoms when hit by high-energy photons or cosmic rays. The result is formaldehyde, CH2O, a molecule that smells terrible but is chemically versatile. Formaldehyde undergoes condensation reactions to form sugars through the Formose reaction. This is step 2, and it has been known since 1861 when Russian chemist Alexander Butlerov first described it. Formaldehyde molecules link together, building longer and longer carbon chains until they close into rings and become sugars. The Formose reaction produces a mess of products, dozens of different sugars in varying yields, but among them is ribose, the 5-carbon sugar that forms the backbone of RNA. In typical for most reactions, ribose is a minority product, maybe a few percent of the total yield, but the breakthrough goes further. Under pulsed irradiation, conditions that mimic cosmic ray impacts or solar flares, methanol can also generate nuclear-based precursors, the nitrogen containing rings that pair to form the genetic code, adenine, guanine, cytosine, uracil. All of them can trace pathways back to simple starting materials like methanol, hydrogen cyanide and ammonia. The pulsed irradiation is key because it mimics the kind of energy delivery you get in space. Not a steady drizzle of photons like sunlight provides on Earth's surface, but bursts, spikes of energy that come from solar flares, cosmic ray hits, or impacts of energetic particles. Those bursts drive reactions that would not happen under steady illumination. Bonds break in specific ways, radicals form, and those radicals recombine into products that are kinetically disfavored under equilibrium conditions. Adenine, for example, requires 5 hydrogen cyanide molecules to link together in a precise sequence. That is statistically unlikely under random conditions, but under pulsed irradiation, with the right mix of methanol, hydrogen cyanide and ammonia, the yield jumps from negligible to measurable. Experimental conditions matter, and the research team was meticulous about replicating space environments. The lab used methanol vapour over icy surfaces, mimicking the conditions on a comet or icy moon. They built a vacuum chamber, cooled a substrate to temperatures around 100 Kelvin, about -173 Celsius, and deposited methanol vapour onto that cold surface. The methanol froze into a thin film, just like it would on the surface of a comet nucleus or in the plume where vapour condenses into ice grains. Then they irradiated it with pulses of ultraviolet and X-rays, simulating the radiation environment in space. Ultraviolet comes from starlight, especially from young, hot stars. X-rays come from stellar flares and from high-energy cosmic events. The pulsing was important. They did not just shine a continuous beam. They pulsed it. Milliseconds of high intensity followed by seconds of darkness over and over for hours, and they found that the yield of RNA precursors was orders of magnitude higher than previous experiments using different starting materials. Previous attempts to make ribose and nucleobases from scratch often started with formaldehyde alone, or with cyanide alone, or with mixtures that did not include methanol. Those experiments produced tiny yields, parts per million or parts per billion, barely enough to detect, let alone call biologically relevant. But with methanol as the starting point, and with pulsed irradiation, the yields jumped to parts per 1,000 for some products. That is a 1,000-fold increase, maybe 10,000-fold for certain nucleobases. Methanol is not just a starter, it is a catalyst, a feedstock and a reactant all in one. It donates carbon, it stabilizes intermediates, it participates in the final steps of nucleobase assembly. It is a chemical multi-tool, and nature, or the universe, seems to use it that way. Direct link to 3i Atlas. The methanol in its plume is not just sitting there, passively drifting through space. It is being irradiated by sunlight, by cosmic rays, by the solar wind. The sun emits ultraviolet radiation constantly, and that radiation hits the plume head-on. Cosmic rays, high-energy protons and heavier nuclei travelling at near-light speed, pepper the plume from all directions. Solar flares, when they happen, send bursts of X-rays and energetic particles that could trigger exactly the kind of pulsed irradiation the lab used. Those conditions could be triggering the same pathways the lab observed. Methanol to formaldehyde, to sugars to nucleobases. The plume is a flying chemical reactor, a distributed laboratory where prebiotic chemistry is happening in real time across volumes measured in cubic kilometres. The output could be far more biologically relevant than we assumed. Think about the scale for a moment. ALMA detected methanol production rates on the order of kilograms per second from 3i Atlas. That is not a typo. Kilograms per second. Over the course of weeks or months of observation, that adds up to tons of methanol released into space. If even a fraction of that methanol undergoes the reactions the lab observed, you're talking about tons of formaldehyde, hundreds of kilograms of sugars, kilograms of nucleobases. Those molecules are not staying put. They are mixing with the plume, condensing onto dust grains, getting carried by the solar wind, drifting through the solar system. Some of them will land on Earth. Some will land on Mars, Europa, Enceladus. Some will escape the solar system entirely and continue into interstellar space. The chemistry of life, or at least the precursors of it, is being manufactured and distributed on an industrial scale by a single object. Realistic counterbalance. Lab conditions do not always translate directly to space, and I need to be honest about the limitations. Concentrations in the lab are higher than in a diffuse plume. The researchers used methanol vapour at pressures that are millions of times higher than what you would find in the coma of a comet. Higher concentrations mean more collisions, faster reactions, better yields. In the sparse environment of space, where molecules are separated by meters or kilometers, reaction rates drop precipitously. Catalytic surfaces, minerals or organic polymers, are often present in the lab but might be absent in space. The researchers deposited methanol onto a silicate substrate, essentially a piece of rock or dust grain. Those surfaces can catalyze reactions, lowering activation energies and directing molecules into specific pathways. If the plume of 3I Atlas has dust grains, and it almost certainly does, then those grains could act as catalysts. But we do not know what those grains are made of, and not all surfaces are catalytic. Time is another factor. The lab experiments run for hours or days. They irradiate the methanol, let the reactions proceed, then analyze the products. In space, reactions might take thousands of years because the energy flux is lower and the molecules are more spread out. But here is the thing. 3I Atlas has been travelling for who knows how long. Hundreds of millions of years, possibly. If it formed in another star system and was ejected during planetary formation, it has had geological timescales to accumulate chemistry. Even slow reactions, given enough time, produce measurable products. And once those products form, they can persist. Space is cold. Molecules freeze onto grains and stay there, protected from further reactions, until something melts them or vaporizes them. So the plume of 3I Atlas might not just be producing nuclear bases now, it might be carrying nuclear bases that formed millions of years ago, stockpiled in its ices, waiting to be released. But the fundamental pathways are the same, and that is what matters. The lab demonstrated that methanol, under plausible space conditions, can make RNA precursors. The details of concentration, surface chemistry and time might differ between the lab and space, but the underlying reactions do not. If methanol can make RNA precursors under any plausible conditions, then 3I Atlas is carrying a payload far more potent than a simple alcohol. It is carrying the raw material for genetic information, for catalysis, for the self-replicating molecules that define life as we know it. Whether those molecules actually assemble into something functional, something that could be called alive, is another question. But the potential is there, undeniable and measurable. Cinematic scenes. Imagine lab footage, the kind of thing you would see in a science documentary. Methanol vapour condensing on a cold surface, frost forming in real time, backlit to show the crystalline structure. Pulses of light triggering reactions, ultraviolet flashes that illuminate the chamber for a fraction of a second, X-ray bursts invisible to the human eye but captured by high-speed cameras, spectrometers detecting formaldehyde, sugars, nucleobases, the data streaming across a monitor, peaks rising on a graph as molecules are identified and quantified, the researchers analysing the results, looking at the numbers, realising what they have found. Then cut to space. An artist's rendering of 3I Atlas, its nucleus dark and irregular, its plume glowing faintly in ultraviolet, a ghostly cone of gas and dust stretching millions of kilometres behind it, molecules assembling in the wake, formaldehyde linking into chains, ribose closing into rings, adenine forming from cyanide and ammonia, overlay with narration. This is not just chemistry. This is the chemistry of life, happening in real time in our solar system right now. Close with the implication. If lab results scale, and there is no reason to think they do not, then 3I Atlas's methanol is far more astrobiologically potent than we thought. It is not just a precursor, a starting point that might or might not lead to something interesting. It is an engine, a self-assembling factory for the molecules that life needs. Ribose for genetic backbones, nucleobases for information storage, formaldehyde for further organic synthesis, all of it streaming out of a single object, distributed across the solar system, seeding planets and moons with the chemical potential for biology. Whether that is accident or design, we still do not know. The lab results do not tell us intent, they tell us mechanism. They tell us that nature, or whoever built this thing, has access to a chemical pathway that is efficient, robust and scalable. They tell us that the distinction between prebiotic chemistry and biology might be thinner than we thought, that the transition from non-life to life might not require a miracle or a statistical fluke, just the right molecules in the right place under the right conditions. 3i Atlas is providing those conditions right now, and December 19th is when Earth might sample the results. One more piece of the puzzle before we wrap up. Mars, dust, organics, and the possibility that our solar system is already a chemically connected ecosystem. Part 15. New evidence of a slow interplanetary microbial drift around Mars. Orbital dust sampling missions around Mars in 2025 detected complex organics in the Martian exosphere, the outermost layer of the atmosphere where particles escape into space. These were not simple hydrocarbons. These were polymers, possibly amino acids, structures that hint at biological or prebiotic chemistry. Whether they originated on Mars, were delivered from elsewhere, or formed in situ is unclear, but their presence suggests something we have suspected but rarely confirmed. The solar system is not a collection of isolated worlds. It is a connected ecosystem, and dust is the connective tissue. Let me break down what this actually means, because the implications are staggering. The exosphere is where Mars ends and space begins, a transition zone so tenuous that molecules can orbit for years before either falling back to the surface or escaping entirely into interplanetary space. Sampling that region requires precision instruments on spacecraft, flying through zones where the particle density is measured in single molecules per cubic meter. The fact that complex organics were found there, consistently, across multiple sampling passes, tells us they are not contaminants from the spacecraft. They are real. They are Martian, or at least Mars adjacent, and they represent chemistry that is actively cycling through the planet's upper atmosphere. The polymers detected show carbon chains longer than 10 atoms, structures that do not form easily through random atmospheric chemistry. Amino acids, if that is what they are, would be even more significant because amino acids are the building blocks of proteins. The analysis is still ongoing, and the peer review process moves slowly, but preliminary data suggest molecules with nitrogen-carbon bonds, chiral centers, and functional groups that match biological or prebiotic profiles. This is not proof of life, past or present, on Mars. But it is proof that Mars is chemically active in ways we did not fully appreciate, and that the boundary between Mars and the rest of the solar system is porous. Interplanetary microbial drift is the idea that microbes, or at least organic molecules, can travel between planets on dust grains or meteoroids. We know this happens because we have Martian meteorites on Earth, rocks blasted off Mars by impacts that eventually fell here. The most famous is Allen Hill's 84001, a meteorite found in Antarctica that some researchers claimed contained fossilized Martian bacteria. That claim is controversial and remains unproven, but the fact that Martian rocks reach Earth is not controversial. It happens. Impacts eject material from Mars, and some of that material, after drifting through space for thousands or millions of years, intersects Earth's orbit and burns through our atmosphere. If rocks can make the journey, so can smaller particles. Dust grains are lighter, easier to accelerate, and less likely to burn up completely during atmospheric entry. A grain 10 microns across, about the width of a human red blood cell, can survive the journey from Mars to Earth if it enters the atmosphere at a shallow angle. Once it lands, it becomes part of Earth's soil, indistinguishable from terrestrial dust unless you analyse its isotopic composition or mineralogy. We have found such grains. Antarctic ice cores contain particles with isotopic signatures that match Mars. Stratospheric balloon experiments have collected dust with mineralogy consistent with Martian origin. The exchange is real, and it is ongoing. If those particles carry organics, or even dormant microbes, then life or its precursors could be exchanging between worlds on timescales of millions of years. Dormancy is key here. Microbes can enter states of suspended animation, where metabolic activity drops to nearly zero. Spores, cysts, and other resistant forms can survive vacuum, radiation, and temperature extremes for extended periods. Laboratory experiments have shown that certain Earth bacteria can survive years in simulated space conditions. Extrapolate that to millions of years, and the odds of survival drop, but they do not reach zero. A tiny fraction of microbes, the most resistant, the most lucky, could theoretically make the trip and remain viable. 3i Atlas fits into this picture as a dramatic example of a common process. Interstellar objects are rare, but they are not unique. Before Oumuamua in 2017, we thought we might see one interstellar visitor per century. Now we know the rate is higher, possibly one every few years if we are looking carefully. Statistical models based on Oumuamua and 2i/Borisov suggest that at any given time, there are tens of thousands of interstellar objects in the inner solar system, most too small or too faint to detect. If even a fraction of those objects carry organic chemistry, as 3i Atlas appears to, then the solar system is being continuously resupplied with prebiotic materials. The supply rate is slow, measured in tons per million years rather than tons per year, but over geological time it adds up. Add to that the interplanetary exchange of dust, and you have a system where chemistry, and potentially biology, is flowing between worlds on a regular basis. Mars to Earth, Earth to Mars. The Moon, Venus, even the moons of Jupiter and Saturn, all are part of this exchange network. Every impact that ejects material from a planetary surface sends particles into orbits that eventually intersect other bodies. Some of those particles re-impact within years. Others drift for millennia, carried by solar radiation pressure and gravitational perturbations, before finding a new home. The solar system is not static. It is a mixing bowl, and the ingredients are being stirred continuously. The timescales matter. A dust grain travelling from Mars to Earth might take 10,000 years. A meteoroid might take 1,000,000. An interstellar object like 3i Atlas has been travelling for much longer, possibly hundreds of millions of years if it was ejected from its home system during planetary formation. But once it arrives, once it releases its payload of methanol and hydrogen cyanide and whatever else it is carrying, that chemistry enters the same mixing bowl. It becomes part of the interplanetary dust population. It lands on Earth, on Mars, on Europa, wherever gravity and orbital mechanics take it. The delivery is slow, but it is persistent. Planetary protection implications are immediate and contentious. If Mars and Earth are already exchanging organics, then contaminating Mars with terrestrial microbes might not be contamination in the traditional sense. It might be acceleration of a process that is already happening. Every Mars rover, every lander carries Earth microbes, despite our best sterilization efforts. We bake the spacecraft, we clean them with alcohol and ultraviolet light, we assemble them in clean rooms, but we cannot eliminate every spore, every resistant bacterium clinging to a circuit board or a cable. Some estimates suggest that each Mars mission delivers thousands of viable microbes to the Martian surface. If interplanetary exchange is already occurring naturally, then our missions are just adding to an existing flow. But other scientists argue that natural exchange and human-caused contamination are different in kind, not just degree. Natural exchange is slow, stochastic, and limited by the harsh conditions of space travel. Most microbes die. Only the most resistant, the most protected, have any chance of survival. Human missions, by contrast, deliver large numbers of microbes quickly, in protected environments like rover interiors or sample caches. The probability that some of those microbes survive and establish themselves is much higher than the natural background rate. If we seed Mars with Earth life, even accidentally, we might irreversibly alter the Martian biosphere, if one exists, or contaminate future searches for indigenous Martian life. The ethical and scientific stakes are enormous. Policy debates on this are ongoing, and 3i Atlas sharpens the questions. International agreements, specifically the Outer Space Treaty of 1967, require that we avoid harmful contamination of celestial bodies. But what counts as harmful? If the solar system is already a chemically connected ecosystem, if interstellar objects are already seeding planets with organic chemistry, then our definition of contamination might need revision. Should we treat 3i Atlas as a contaminant? Should we try to prevent its chemistry from reaching Earth? The practical answer is no, we cannot prevent it. The object is too far away, moving too fast, and dispersing molecules over volumes too large to control. But the philosophical question remains: do we have a responsibility to monitor it, to sample it, to understand what it is doing before it does it? Monitoring strategies are within our capabilities. Atmospheric sensors, ground-based and space-based, can detect changes in trace gas concentrations. If methanol or hydrogen cyanide from 3i Atlas reaches Earth's upper atmosphere, We should see spikes in those molecules in the days or weeks following December 19th. Spectroscopic surveys of the exosphere, the same kind that detected organics around Mars, could be deployed around Earth. Dust collectors on high-altitude balloons or on satellites could capture particles from the wake. Isotopic analysis of those particles would reveal their origin. If they match 3I Atlas's composition, we have confirmation of delivery. If they do not, we know the object's chemistry dispersed too widely to reach us in detectable amounts. Sampling the wake directly would require a spacecraft intercept mission, and there is not enough time to launch one before December 19th. But future interstellar visitors could be targeted. Concepts exist for fast flyby missions that could rendezvous with incoming objects, deploy instruments, and return data or even samples. The technology is proven. We have flown past comets before. Stardust mission collected samples from Comet Wild 2. Rosetta orbited Comet 67P and deployed a lander. Doing the same for an interstellar object is harder because of the high relative velocity, but it is not impossible. If we prioritise it, if we build the hardware and keep it ready to launch on short notice, we could catch the next 3I Atlas and answer these questions definitively. The Mars findings suggest we should. If organic polymers are drifting through the solar system, if amino acids are cycling between planets, then understanding the sources of that chemistry becomes a priority. Is it mostly asteroidal, material from broken up planetesimals? Is it cometary, volatiles released from icy bodies? Or is it interstellar chemistry delivered by visitors like 3I Atlas? Each source has different implications for the origin and distribution of life. Asteroidal organics are ancient, preserved from the solar system's formation. Cometary organics are dynamic, released and reformed as comets orbit. Interstellar organics are exotic, carrying the chemical fingerprints of other stellar systems. Distinguishing between them requires detailed isotopic and molecular analysis, the kind we can only get from samples. The bigger picture is this. We live in a solar system that is not closed. It is open to exchange, both internally between planets and externally with the wider galaxy. 3I Atlas is a reminder of that openness, a dramatic, visible, chemically active reminder that the boundaries we draw on maps and in our minds are not real. The solar system is a node in a galactic network, a waypoint on chemical pathways that span light years and eons. Whether those pathways carry the seeds of life, the poisons of death, or just the random detritus of cosmic evolution, we do not yet know. But we are learning, and 3I Atlas is one of our teachers. End the series with the big picture. 3I Atlas is a door to debate, a gateway to questions we have avoided because they seem too strange, too distant, too speculative. But the data forces us to confront them. Natural wonder, hazard or invitation. We do not yet know which. The chemistry is calling us to look harder, to think deeper, to prepare for possibilities we have not imagined. We do not yet know whether we will be fertilized or fumigated. We only know that something is coming, and December 19th is when we find out. And maybe, just maybe, the answer will not be binary. Maybe it will be both. Maybe the universe does not deal in simple categories, in gardeners versus killers, in friends versus enemies. Maybe it deals in chemistry, in molecules that carry potential, in reactions that unfold over timescales longer than civilizations. Maybe 3I Atlas is neither saviour nor threat. Maybe it is just a comet, doing what comets do, venting molecules into space because that is what happens when ice meets sunlight. Or maybe it is something else entirely, something we have never encountered before, something that will redefine what we think is possible. Either way, we are about to find out. December 19th is not the end of the story. It is the beginning, and we should be watching.